Hello and welcome to our BIPOC Neuroscientist Spotlight. My name is Isobel Matsukas and I work with Christine Salcedo, Ali Sereno, and Maria Cortez-Becerra. The first scientist we're going to talk about today is Emery Brown.
Emery Neal Brown was born in Ocala, Florida in 1957, and both of his parents were math teachers. In 1974, he graduated from Phillips Exeter Academy after studying abroad in Barcelona, Spain. 4 years later, in 1978, he graduated magna cum laude from Harvard College with a degree in applied mathematics and accepted a fellowship at Institut Fourier de Mathématiques Pure in France. After his fellowship in France, Brown returned to Harvard Medical School and entered the MD-PhD program and earned his MA in statistics from Harvard University in 1984, his MD from Harvard Medical School in 1987, and his PhD in statistics from Harvard University in 1988. Brown completed his internship in internal medicine at the Brigham and Women's Hospital in Boston in 1989. By 1992, he completed his residency in anesthesiology at the Massachusetts General Hospital, or MGH. After completing his residency, he joined the Department of Anesthesia at MGH and the faculty at Harvard Medical School as an instructor. In 1998, he joined the faculty of Massachusetts Institute of Technology, or MIT, as a professor of computational neuroscience and professor of health and sciences. Currently, Brown is both an Edward Hood Taplin Professor of Medical Engineering and of Computational Neuroscience and a Professor of Health Sciences and Technology at MIT. He is also a Warren M. Zapple Professor of Anesthesia at Harvard Medical School. He serves as the Director of the Harvard-MIT Health Sciences and Technology Program and the Associate Director of the Institute for Medical Engineering and Science at MIT. Lastly, he is an investigator at the Picower Institute for Learning and Memory in the Department of Brain and Cognitive Sciences at MIT. He is a fellow of several organizations, including the American Institute for Medical and Biological Engineering, the American Statistical Association, the American Association for the Advancement of Science, and the Institute of Electrical and Electronics Engineers. Brown is also a member of the National Academy of Science and the Institute of Medicine.
Brown conducted research and work with statistics since he has a PhD in that field, but we focused on his neuroscience research for the sake of connecting it to our course. Understanding how the brain transmits information, firing patterns, characterizing the spiking properties of neurons and neuronal activity during differing arousal states, and effects of anesthesia on the brain are all focuses of his research. Scalp EEG recordings are used to measure changes in brain states under anesthesia, which is one of the research experiments that he conducted. Global coherence is the ratio of the largest Eigen value to the sum of Eigen values of the cross-spectral matrix at a certain frequency and time. This is used to analyze the spatiotemporal dynamics of multivariate time series. He found that during unconsciousness, there are increasing alpha and delta powers, and the occipital has more delta power than alpha power. He also found that more alpha activity in occipital during consciousness, which shifts to the frontal during unconsciousness. Brown also studied sleep. Sleep is generated by nuclei in the hypothalamus, brainstem, and basal forebrain. In REM sleep, REM stands for rapid eye movement, the EEG gives a high frequency and low amplitude graph, while non-REM sleep has 3 stages with higher amplitudes and lower frequency rhythms. PPT/LDT neurons influence REM sleep initiation but are not active during REM sleep duration. Anesthesia acts on the central nervous system, and the targets differ depending on what kind of altered state of arousal is trying to be obtained. Anesthetic drugs act as agonists on GABA A receptors because small doses induce sedation. He also studied stimuli, and he used electrodes to do so. The use of electrodes to study the activity of neurons in a specific region of the brain, specifically. A working memory task gives way to neural activity in the hippocampus or prefrontal cortex, and then the light stimuli on the retinas or lateral geniculate neurons. Then the voltage signal spikes, representing an increase in neuronal activity. Lastly, connections to our course. We are currently learning about action potentials within the neuron, and this is directly related to how the brain transmits information, firing patterns, and the spiking properties of neurons. We also learn how the brain transmits information in relation to behavior, and we learn about how drugs can influence the brain, specifically anesthesia. Another component of this course is learning about research techniques and tools which Brown implemented in his research, the EEG recording, for example. He also used electrodes and voltage signal graphs to research where neuronal activity takes place in the brain based on certain stimuli, neuronal activity in the retina from light stimuli, for example.
The second scientist we researched is Mahmood, Reza, Amiry-Moghaddam.
Mahmood Reza Amiry-Moghaddam is a Norwegian-Iranian neuroscientist born on April 21, 1971. He studies many things in his field. Some of his studies and interests include the brain and nervous system, cell volume regulation, aquaporins, astrocytes, and neurological disorders. Outside of neuroscience, he is also an Iranian authorities human rights advocate. He got his MD at the University of Oslo in 1996 and got his PhD in 2003 at the same university. He went on to the Department of Developmental Biology for his postdoc and studied at Harvard School of Dental Medicine in 2006. Lastly, in 2009, he became a professor in medicine and a group leader at the Laboratory of Molecular Neuroscience in the University of Oslo. Mahmood started his interest in neurosciences his second year as a medical student. He started his work from studying kidneys and moved on to aquaporins shortly after the aquaporins water channels were discovered. He then switched his focus to neuroscience. Neuroscience is an area of study that Mahmood is fascinated about. He seeks to discover more about the brain, especially brain disorders. He also focuses a lot of his time researching glia cells such as astrocytes. His main purpose of evoking so much time onto glia cells is that neurons have been the main focus for centuries, potentially being the reason as to why many brain disorders have gone uncured. As previously mentioned, Mahmood is also an Iranian authorities human rights advocate. His main drive to becoming active in politics was his upbringing. Growing up, Mahmood saw the voices of the brave become silenced by people in power. Political views were questioned and enforced onto everyone, even children. Children were questioned on what was happening at home for the sole purpose of ensuring that political views remained where they should be. In 1981, mass public execution became very common, including one of his cousins of age 18 being executed for supporting an opposition group. Seeing this at a young age influenced him to speak up, and he quickly realized that he can stand up all on his own. His long-term wish in being an Iranian human rights advocate is to abolish the death penalty. Many innocent people are being executed every day due to political affiliations.
The majority of Mahmood Amiry-Moghaddam's work relates to glial cells. Amiry-Moghaddam's work relates to the role of glial cells in water transportation, postnatal development of astrocytes, and the connection between glial cells and brain aging. The role of glial cells in water transport led to the discovery of glial cells possessing endocoding aquaporin 4, AQP4, in their cell membranes. Therefore, glial cells are specialized for water transport and play a large role in mediating water flow between areas with CSF and the intravascular space. This relates to our Neuroscience 175 course content because we discuss the unknown possibilities of glial cells when we discuss neurons. Amiry-Moghaddam's work allows us to see the capabilities of glial cells because he is one of the scientists who is conducting further research on them. Research into the postnatal development of astrocytes has created an interest in studying extracellular matrix, ECM, in relation to neurological diseases. Amiry-Moghaddam discovered a connection between ECM and the polarization of astrocytes. The loss of polarization of astrocytes causes some neurological diseases, therefore establishing a link between astrocyte polarization and ECM also establishes a link between ECM and neurological diseases that needs to be further explored. This relates to our Neuroscience 175 course content, because we discussed the specific roles of astrocytes in the brain and we learned how astrocytes communicate with each other through their polarization. The connection between glial cells and brain aging led to the discovery of a connection between glial cells and exercise. Amiry-Moghaddam concluded that studying exercise through its effects on glial cells could lead scientists to be able to detect aging-related brain dysfunction earlier than they previously could. This pertains to our Neuroscience 175 course content because it again discusses the importance of glial cells, and Amiry-Moghaddam is a prime example of a scientist who is further looking into glial cells and their capabilities.
Thank you for watching and following our sources and our image sources. Thank you and have a great day.